for in vitro tissue models. And to quickly run through our presenters, our first presenter is currently showing his face, Dr. Min-Jae Song, who is employed at the National Center for Advancing Translational Scientists, otherwise known as NCATS. He specializes in the development of 3D vascularized tissue models within a high throughput platform designed for preclinical studies. So we're looking forward to having him present first. Our next presenter will be Dr. Tuan Hoang. Dr. Tuan Hoang is employed by Finovance and oversees their chip production and development laboratory. He is an accomplished industrial bioengineer and a recognized expert in microtechnology. And our last presenter is Dr. Patrick Hayden, who works for Bioservices and is an expert in tissue models and developing those out. He also holds a position here of overseeing the design and direction of studies related to the interaction and growth of human tissue cells within electrospun nanofiber materials. So we look forward to those presentations today. Just to run through the agenda real quick, I think we set it for around 2.5 hours. We were hoping that it will go by a little bit quicker. We just figured in case there was a lot of questions, we might as well extend it out. So as I already mentioned, the webinar and the speakers. So our first one will be Dr. Min-Jae Song, and he'll go over the predictive human in vitro models for improving translational quality and efficiency. So I will let MJ share his screen now, and we can get started.
Thank you very much, Cameron. Let me share my screen. Is it possible to? Can you see my screen, Cameron?
Yep, we can see your screen, and if you need to use your mouse, we can see the mouse as well. So.
Oh, okay, got it. So what kind of screen are you seeing now? The full screen?
Yep, the first screen right now, currently.
All right. Let me change the pointer option to the laser. So you can see it now here, right?
Yep.
Okay. So thank you very much for the introduction, Cameron, and thank you very much for organizing this webinar. And I'm really happy to present the works that we have been doing at NCATS Tissue Bioprinting Lab. And my presentation will include the complex in vitro model and to model the tissues and diseases in the high throughput platforms for drug screening, and also the brief introduction of this NCATS 3D Tissue Bioprinting Lab. So as you know, the over 90% of the developed drug have been failed during the clinical trials for the last 2 decades, and the low predictivity of the current assay platforms is thought to be one of the main reasons for this high failure rate. So this translational gap between the basic science and clinical science is very challenging roadblock due to the high risk of the failure and taking the high cost, which is called the valley of death. So as you can see in this chart, the translational steps take over the 80% of the entire cost of the drug development, and then this failure rate remains not improved, although the drug development cost is annually increasing. So as a government agency, the NCATS tackles these translational problems to improve the translational quality and efficiency. And doesn't. Okay, sorry. Recently, the several innovative in vitro models were developed and demonstrated better the physiological relevance, and then the models have been already extensively used in the basic science field. And so accordingly, the NCATS established the 3D tissue bioprinting lab to implement the 3D models into the translational sciences to improve the predictivity of the assay platforms for drug screening, for drug discovery. The NCAS 3D tissue bioprinting lab was initiated in the 2014 to learn the 3D cell culture technique starting from the skin and the ocular tissue models. And then these established protocols from these models were further expanded to the other tissue models and the digital models. And our lab became an official laboratory in the 2018, led by Dr. Mark Ferrer. So NCAT's 3D tissue bioprinting lab is aiming to the biofabricate and then validate these 3D tissue models with robust assay readouts in a high throughput platform for drug discovery. And as I mentioned here, I call the concept of our work is the translational bioengineering, meaning that we are not developing the bioengineering technology itself, unless it is necessary. So we'd rather to investigate the current state of the art and identify the most effective platforms and then engineering solutions for the project. So we translate the bioengineering the technologies into the drug development processes by the combination of the 3D cellular models, 3D cell 3D cellular models here, and then also the engineering solutions such as the bioprinting technology and also organ-on-chip technology. In the basic science, the physiological relevance and complexity is the priority, while the level of throughput is the low priority. Screening of the hundreds and thousands compound is not necessary. On the other hand, the level of throughput is the critical in the drug discovery process, and then accordingly, the reproducibility is a key for successful translation. So hence, the ultimate goal of our lab is to achieve the 3D in vitro models in a high throughput platform with acceptable the physiological complexity depending on the context of use, eventually helping the reaching the value of death. This figure just shows the overall steps of the developing the 3D models. Once we determine the tissue and the disease models, we search for the working cells that express the biomarkers and then meet the tissue-specific functional outcomes. So there are three different kinds of the commercially available cell sources, as you know, like the human primary cells, immortalized cells, and then iPS-derived cells. The human primary cell source is, of course, the most physiologically relevant, but the limited lifespan of the particular cell types became the challenges for the large-scale screening project. The immortalization technique has been developed, but it has not been achieving the physiological relevance yet. And the human iPSC technology hold the strong benefits by meeting both this physiological relevance and then also scaling up. However, the cell sources are dominantly limited by the quality of the differentiation protocol. So for this reason, we prioritize using iPS derived cells, but we have expensively used the human primary cells for the most of our tissue models. So once we determine the cell types, we optimize the hydrogel and also the hydrogel materials and also culture conditions. And for the validation of the 3D models, we confirm the structure and function of the model first. Then we check that the cytokine secretion, also the transcriptomics and also the pharmacological outcomes using the clinically tested compound with the non-clinical outcomes. For high throughput assay development in the 3D, it is often challenging to implement the technologies that were used for the validation of the 3D models into the high throughput assay development due to the time sensitivity or the hardware complexity. Finally, the data management is demanded as data size and amount are huge from the high throughput assays. So once this individual step is achieved, it will become a high throughput assay ready platform. So by the way, the most challenging part here that we have been facing now are the identification of the good cell sources and the developing the high throughput functional measurement. And then I can say that the most half of my time is to identify the cell sources for individual tissue models. So the next slide is, this figure shows the portfolio of the tissue models that we have been developing. And as I mentioned that we have started the ocular tissue and the skin tissue models in 2014. And the ocular tissue project was initiated under the collaboration with the NEI, National Eye Institute, um, to develop the outer blood retina barrier tissues to model the age-related macular degeneration. So ICA recently published in demonstrating the choroidal neovascularization-like morphology, which is the hallmark of the web form age-related macular degeneration. And then I will show more details in the following slides. We established the different levels of the complexity of the skin tissue models, and then the second figure shows that the 3D vascularized full thickness skin models here, skin models in a transfer format using the bioprinting technology. And using this model, we published a atopic dermatitis model with the JAK inhibitor treatment. Similarly, the lung model also has different levels of the tissue complexity. As COVID-19 pandemic began, that we initiated in the lung tissue model project in a both Transwell and the organ-on-chip platform. And then the later slide, I'm going to show you the organ-on-chip platform of the lung tissue model, which was published in the biofabrication paper in the 2020. A couple of years ago, we launched a project to develop the placental barrier model to study the fetal toxicity of the drugs and the viral infection. And then we also developed a vascular unit with the astrocyte to develop an assay for the glioblastoma, which is the brain tumor, within the context of the vascular unit. As a part of the HEAL program, which is the Help to End Addiction long-term program, we have been developing a neurocircuit model to assess the pain signal of the neurons in a high-throughput platform. And we have been developing also a vascularized bone model to study the wound healing process after the bone fracture under collaboration with the NIDCR. And then finally, we developed the skeletal muscle model using a unique commercial platform, enabling the simultaneous measurement of the contractility in a 24-well format. And this model will be further applied for the increasing the complexity to achieve the neuromuscular junction model by introducing the iPS-derived motor neurons with collaboration with the NINDS. And also we eventually use this model for the gene therapy application and then modeling the rare diseases. So now that the later slide, I'm going to concentrate on only the focus on only the retinal tissue and the lung model. However, if you are interested in the other models, just please let me know. We are happy to discuss further. So these two models are the platforms that we have been currently using, which is the left-hand side is transfer platform using the bioprinting technology, and the right-hand side is organ-on-chip platform to enable the perfusion of the 3D vascularized tissue in a high-throughput platform. And then just brief introduction of this transfer platform with the bioprinting. We simply mix this endothelial cell, fibroblast, and pericyte, which are the key components of the vascularized tissue, embedded in the hydrogel, which we call the bioink. We loaded this bioink into the syringe, and then we put this syringe into the printing head of this bioprinter. So using the extrusion base of the bioprinter, we printed this bioink in a certain pattern of in a designed geometry in the bottom side of the transfer membrane, and then we flip it back, and then we put the epithelial cell on the other side of this transfer membrane to complete the basic structure of the barrier tissue with the vasculature. And then using this protocol, we applied the organ-on-chip platform to perfuse this vasculature as well. And then now I'm starting to explain about these ocular tissue models to model age-related macular degeneration. So just the brief background of these ocular tissue models, and then we developed this outer blood retina barrier, which is located in the back of the eye, and then in the back of the eye is composed of the different layer of the tissues, which include the neurosensory part of the retina and the photoreceptor, and then RPE, which is the retina pigment epithelium, which is a homeostatic unit of the back of the eye to maintain this viability of this photoreceptor and the choroid. And then underneath of this RPE, there is a special the basement membrane of the basement membrane, which is called the Brooks membrane. And then underneath of the Brooks membrane, there is a highly vascularized tissue, which is called choroid. So we to model the age-related macular degeneration, we model this outer blood retina barrier, which composed of the RPE and also the Brooks membrane and choroid, which contains highly vascularized tissue. And there are two different kinds of the AMD. The first one is the dry form AMD, and then the second one is the wet form AMD. And the dry form AMD, the degeneration of the RPE causes the cell death of the cell death of the photoreceptor, and then also the degeneration of the choroid causes the irreversible vision loss. And then wet AMD, on the other hand, during the development of the wet AMD, malfunction of this RPE causes excessive blood vessel growth from these choriocapillaries toward this RPE, and then through the disrupted this RP barrier, the excessive blood vessel growth eventually disorganize this photoreceptor to cause the irreversible vision loss. And this excessive blood vessel growth is called the choroidal neovascularization, which is called CNV in the pathological condition. And then right-hand side videos just show that there's 3D models of the wet AMD model with the RPE containing RPE with the vascularized tissue. So first of all, we developed this vascularized tissue using the bioprinting technology in the transfer platform. And then as you can see, I already explained a little bit about this process of the endothelial cell fibroblast and pericyte mixture. And then I'm going to just skip this one. And the left-hand side bottom shows that this video of the bioprinting with the stripe shape of pattern of the bioprinting on the bottom side of the transfer membrane. And the right-hand side, the image shows that the JP positive endothelial cell making the tube formation within the stripe shape of the printed structure. And then after that, they are branching out into the empty hydrogel, which we called it angiogenesis. The benefit of the printing this certain shape of this vascularized tissue is facilitating the image quantification of the pure angiogenesis, which is the branching out from this printed structure. And then in here, especially we use this biodegradable polymer scaffold generated by the electrospinning technology. And we simply replace the PLG non-fiber scaffold with the commercial the transfer membrane here. And then benefit of the PLGA transfer membrane, PLGA, the biodegradable polymer scaffold, is to increase the tissue integrity by degradation of this scaffold, and also the degraded space of this scaffold maximize the communication between the epithelial cells and also the vascularized tissue as well. So this one is showing the morphology, the validation of the morphology by staining of the CD31, which is the endothelial cell marker. And we confirmed that the tube formation morphology by testing 3 different kind of cell lines, such as the JP positive endothelial cell, and also the human primary endothelial cell without any fluorescence from the different donor. And also we confirmed that this tube formation using the iPS-derived endothelial cells as well. And also we include, as you know, that we included the pericyte here, that the role of the pericyte is the stabilizing the vascular morphology. So and then we confirm that this parasite is surrounding this vasculature, which is mimicking the in vivo condition in that this TEM image showing that the parasite is surrounding this vasculature. And also we confirm that the parasite is attaching to this vasculature by staining of the parasite marker, which is NG2. And we also confirm the endothelial cell tubes are the hollow tubes by the histology and also the vibrotom cut with immunostaining with the CD31, which is endothelial cell marker, and the NG2, which is the parasite marker as well. So using this vascular bed, we added the RPE on the top, on the other side of the scaffold. And then we confirm with the different kind of the markers here. So left-hand side figure is ezrin. As you can see, that ezrin is expressing on the RPE, and the E-cadherin, which is a junctional marker of the junctional marker of the epithelial cell, expressed in the RPE as well. And then when you see this as an expression on the apical side of the RPE, which provide the evidence of the polarization of this RPE as well. And then we confirm this the PRBAP, which is a fenestration marker of the endothelial cell, which is a special feature of the choriocapillaries. And that this fenestration of this choriocapillaries is accelerating the gas exchange between this blood side to the RPE side. So we confirm this expression of PRBAP. However, this expression of the PRBAP doesn't necessarily mean that the fenestrate is forming in the actual endothelial cell here inside of the tissue. So we confirm the fenestrate formation, the ultrastructure of the fenestrate formation in this endothelial cell tubes by the TEM electron microscopy as well. We also evaluated this barrier function of this RPE by using this TEER measurement, which is transepithelial electrical resistance measurement. It basically measures the electrical resistance between the apical side of the RPE and the basal side of the choroid. And then we observed that this inclusion of RPE is dominantly increased these TEER values inside TEER values of the outer blood barrier tissues. And after confirming the structure and function of engineered outer blood barriers, we also studied the single cell RNA, single cell transcriptomics of the outer blood barriers. And as you can see here, the green color is the two-dimensional, and then also the brown or the orange light color is the three-dimensional tissues. And this and then this purple cluster is showing that the three-dimensional tissues with RPE. And as you can see, the two-dimensional and the three-dimensional shows the different pattern the different patterns of this transcriptomics here in the RPE and also this endothelial cell, fibroblast, and parasite as well. And then furthermore, the inclusion of RPE also they have the different cluster of this transcriptomics comparing to these three-dimensional tissues without RPE as well. So I'm going to show you the little bit of the detail of these endothelial cells with the comparison between the 2D and 3D, and also the comparison between the RPE and the without RPE inclusion. So here, between the 2D and 3D, we observed that the cell cycle and the tumorigenesis related genes were downregulated in the 3D comparing to the 2D. On the other hand, the EC maturation marker is upregulated in the 3D. And then by inclusion of the RPE, we further saw that there is more the EC the maturation markers maturation markers were upregulated, And also, if we look at that, this TTR is the protein secreted by the choroidal endothelial cell, which is more specific to the outer blood-brain barrier. So inclusion of the RPE stimulated this RPE secretion of the endothelial TTR protein secretion by endothelial cells. And then we also confirmed that the RPE choroid homeostasis-related genes were upregulated, and also the Brooks membrane gene related gene markers were upregulated by inclusion of the RPE as well. So using this model, we further developed these dry AMD models by treatment of the complement competent human serums. Because the complement pathway has been a key of this developing of the dry AMD. So we treated the human serum treatment, and as you can see in the top row, this is control, which is quote unquote the healthy model, and then the bottom row is showing this complement competent human serum treatment. And as you can see, in the very first column is right below this the RPE, and the second column is the RPE region, and then third column is choroid region. And we by staining of the apoE, and there is a lot of the lipid accumulation right underneath of the RPE by treatment of this complement competent human serum, and also we saw the disrupted RPE layer and also degenerated choriocapillaries, and which is mimicking the the dry AMD progression. And we also confirm that the barrier function is decreased by the treatment of the human serum as well. And then we also confirm that the another lipid accumulation, this lipid accumulation, the right underneath of the RPE by staining of the nylate as well. And then we also modeled another AMD, which is the wet AMD model. Then during the development of the wet AMD, the hypoxia is the key initiator of the AMD. So we treated this HIF-1 alpha inducer to only the apical site of the RPE here. So by treatment of the inducer to the apical site of the RPE, we confirm that there is an excessive blood vessel growth toward this RPE, which mimics type 1 CMV. And we also confirm that decreased the barrier function by treatment of the HIF-1 alpha inducer. And then anti-VEGF is a standard treatment of the wet AMD in the clinics, and it has been showing the rescue of this wet AMD the CMV pathology in the wet AMD development, and then it just hinders the progression of the wet AMD. So we also confirm that by treatment of this anti-VEGF on the top of this RPE, on the apical side of the RPE, showing that the decreased the type 1 CMV here as well. And also we saw the partially improved rescued these barrier functions with treatment of the anti-VEGF. So in summary, in the ocular tissue model, we developed the 3D models of the outer blood-brain barrier, quote unquote, the healthy model. And then based on this healthy model, we developed the disease model of the wet AMD and also dry AMD model. And then we tested this anti-VEGF for the pharmacological validation. So let's switch the gear a little bit. I think I have a little bit of time that I will try to finish this presentation in the 5 minutes. So this long model has the 4 different kind of the complexity here, and the very left hand side is the simplest model, just adding the epithelial cell and the second column here is the printing the vascularized tissue without epithelial cell. And then combination of the vascularized tissue and then epithelial cell together, we have been developing this vascularized full thickness long model in the transfer format, like very similar to this ocular model as well. And then recently we have published this the organochip platform model to model the lung model in a with the perfused this vascularized tissue in 2020. And then we simply just using the the optimized protocol of the lung endothelial cell, lung fibroblast, and the lung pericyte in a optimal hydrogel, and then we put it in here, and we successfully perfuse this vascularized tissue with epithelial cell. So once we establish these models, we characterize this the morphology, and also the morphology with the staining with the markers. Also, they evaluated the barrier functions such as cell viability, and also the barrier function and perfusion assay as well. So this is the organochip models in a high throughput amenable platform. There are 64 tissues possible in this in the one platform, and the individual unit has the three different channel here. And then left-hand side channel is a left-hand side channel, and the right-hand side channel is adding this endothelial cell monolayer. And then the middle channel is the channel that we are loading this hydrogel mixture with the endothelial cell, fibroblast, and pericyte. And then as time goes by, they are making the vasculogenesis through this microvascular network. And then we added this epithelial cell on the top of this vascularized tissue to complete the basic structure of this vascularized lung models here. And this figure shows that at the day 7 after loading this hydrogel, and we simply added the 70 kilodalton of dextran in the left-hand side channel, and we observed the 70 kilodalton dextran got perfused through the microvascular network toward this right-hand side channel here. And then we confirm this morphology of the vasculature with the biomarker of the endothelial cell, which is a CD31. And we also confirm the biomarker of the alveolar type 1 and type 2 cell markers, such as the caveolin 1 and the ABCA3 markers as well. And we confirm that the markers of the small area epithelium, which is the epithelial cell, which is ciliated marker, the alpha tubulin, and also globular cell marker, which is a MUC5AC, and then basal cell marker, which is a CK5. And we also confirm that inclusion of the alveolar epithelium or the small area epithelium that dominantly increase the barrier function, and also with inclusion of the epithelium and vasculature synergistically increase this barrier function in this model as well. So we have been optimizing the tissue complexity in a high throughput platform depending on the context of use, and then complementing the cutting edge technology to validate the tissue models from the molecular level to the tissue levels. So for applications of fully validated these 3D models to the high throughput assay development bring the challenges in the both technical development and clinical relevance. For technical challenges, of course, I previously mentioned the reproducibility of the model outcomes. Reproducibility of the model is also the challenging, and at the same time, the limitation of the current technology hinders the high throughput measurement of the time-sensitive functional outcomes of the 3D tissue models, such as the vascular permeability and the neuronal activity and the muscle contractility. So as 3D tissue model is sitting right between the 2D monoculture and the 3D native tissue, the assay readouts from the tissue model need to be carefully evaluated by bridging studies with the clinical data. So as this effort required a broad range of the expertise, we are bringing the knowledge and experience through a number of collaboration with the basic science field from the NIH intramural and the extramural institutes, and then drug developers from the industries, and of course, FDA. And I want to specially thank to the Mark Farrer, who is the director of the 3D tissue bioprinting lab at NCAT, and also the specially thank to all the long team and also the NEI teams as well. Thank you very much. And if you have any questions, to further, there is my contact information there. So please feel free to just contact me for any questions. Thank you very much.
Awesome. Thank you so much, MJ. Anyone, if you have any questions, you can put it in the Q&A section. I noticed we already have a few questions, MJ, so I'll go right ahead and get started on the first one.
Awesome.
The first question is, blood vessels are composed of EC and pericytes. Why do you include fibroblasts? What is the source?
So fibroblast is the most also very important for the the vascular formation, because the fibroblast is the main source of the developing the extracellular matrices as well. Then they are also the main one of the main sources secreting all the pro-angiogenic factors as well. So fibroblasts need to be included. It is very essential, the cells, for developing this 3D vascularized tissue. And the source of the fibroblast is primary human primary fibroblast. It depends on the the what kind of cells you're using. So for example, this ocular tissue model, the fibroblast is from the human derived, the human donor derived the fibroblast. And also this for the lung model that we use is commercially available, this the human primary fibroblast as well, lung fibroblast as well.
Awesome, thank you. Hopefully that answered your question, Horace. The next question is kind of a three-parter. So, how important is the PDLGA scaffold for successful creation of the retina model? Are there specific properties required? And is this commercially available, or was this custom made for NCAS?
Well, we started this PLGA model because the NEI, the Kapil Bharti, Dr. Kapil Bharti's lab at the NEI, they uses this scaffold model for the transplantation purpose. And then they have been developing this PLGA, and then there is a certain property of the degradation for this translation that all the details maybe you can read Dr. Bharti's papers more, and then they will just describe all the details about this property. And for the development of these ocular models that based on ocular in vitro models to develop this the wet AMD, the degradation is the critical, because if we use this commercially available this PET membrane, which is known to the non-degradable the transfer membrane, because of pore size, you cannot. It is very challenging to develop this kind of the wet AMD morphology, because the blood vessel cannot go through this the quote unquote the membrane, cannot go through this membrane. But if you have the good control of the degradation of this PLGA, then if you have the fast degradation of the PLGA, then this degraded space kind of the wide open, and then this vasculature is go through this this kind of the degraded space toward this RPE. So this is mimicking this the pathological condition of this wet AMD. It really depends on what you want to do. But if you see that there is a no like cell to cell migration, or the no like the usage of this kind of the degradation property, then probably it's okay to use the transfer membrane. But another one is another benefit of the biodegradation is it really help the tissue integrity. They are making all together. So what we experience about this transfer membrane is, if we develop this commercial transfer membrane together, and then if we use our method, then when you do the histology, what we often experience is the after fixation, and then when you punch them out, they kind of easily separated it out. So that means that the integrity of the tissue is kind of weak, and then they are barely attached together. But using this the PLG scaffold is really, they are all just tightly together. So when you punch them out, and they are all just together. And that's why we try to use the biodegradable scaffold. And the tricky part is, it is very hard to control the degradation. So about the commercial product, I guess, there are there are the commercial products here. Here the biosurfaces as well. They have the commercial product developed here in the Transwell, and then I believe that there are other the commercial product of this biodegradable polymer in the multi-wear platform as well. So hopefully this helps.
Yeah, that was great. Thank you for the in-depth answer.
Yeah.
So I think this is the last question here. And then if anyone else has any more questions, don't be afraid to ask, or I'll send out an email and with MJ's information too that you can contact him through. The last question is, what are current limitations to the models you are developing, such as cell source, scaffolds, and bioprinting options?
The cell source is the very, very challenging because first of all, the developing this kind of the in vitro models, that you need a lot of cells, and the primary cells usually, the endothelial cell fibroblast probably okay because you can passage them, but the epithelial cell, for example, epithelial cells, they are not proliferating really well. And when you develop it, for example, for the lung epithelial cell, when you're developing it, you know that cells are very, they're making the barrier function. But once in the middle of the developing these models, if you change the donor, then oftentimes actually it doesn't really work for you. You need to re-optimize all the media conditions and also the cell culture conditions, like as well. So it is very challenging. So that's why we prefer to use the iPS derived cells. But another one is the differentiation. It really depending on the differentiation quality. Sometimes it works, sometimes it doesn't work, and then also the donor dependency just kicking in, and then it is again very challenging to achieve this platform as well. And then scaffold wise, as I mentioned it before, the degradation is tricky. Sometimes, the batch differentiation, the batch to batch difference as well. And then there is the tricky develop this, high throughput platform in a 96 well, such as 96 well. And then sometimes, some well is kind of, the fast degraded, and then, that they are tear the tear earlier than others, and then it is hard to control this uniform degradation of the scaffold in a transfer. So yeah, I think that we need to have the better control of this technology as well.
Great. Well, that looks like, I think it's it for the questions. I really do appreciate you for coming on the webinar, MJ. That was extremely interesting, and hopefully more questions will be coming your way through email. Thank you so much for presenting.
Thank you.
So, all right, so I'll share the agenda just quickly. Next, we will have Tuan present, and then Patrick Hayden will be last. If you guys did miss anything, or you kind of want to go back and rewatch potentially something, this will be posted to our website eventually. So give me 2 seconds to make sure I give this agenda. So as I mentioned, Tuan will be next. His talk will be about humanized models for human applications and progress and advanced 3D cell culture models. So I'll let Tuan take control, and then Patrick Hayden will be the last presentation.
Yes, thank you, Cameron. I'm sharing my screen now. I hope it's starting now.
Yep.
Is everything good on the other side? Okay, good to go. Yeah. Thank you, everybody, for having me today. So I'm Tuan from FinAdvance, and my talk today will be briefly discussing about the Akita platform. So Akita is a product line made by FinAdvance. We are a biotech startup from Oulu, Finland. We started the company 4 years ago, since 2019, and up until now we accomplished already more than 20 patents. And they are now pending or granted all over the world, the US, Europe, and we secure funding from VCs, EU granted projects, and also state fundings. And with 12 experts in the team, ranging from engineers, scientists, doctors, and also business developers, we aim to bring the technologies of microfluidics and organ on chips to accelerate the preclinical drug testing. So with that, we aim to have a high throughput and scalable in vitro model to support the new drug therapies development and bring it faster to the market, and also to improve to really aim for personalized medicine. So about Akita platform, so what we do, we basically have the flagship, which is the well-played format, or we can call it standardized ANSI/format of 96 or 384, would include from 32 to 128 units of microfluidic channels in packing inside the place. And with that, we are able to perform or to mimic different organs, human organs on chip models. For example, vascularized barriers, organoid tissue cultures, endothelial or epithelial barriers. And I will go into a bit more detail in the later slides about those models as well. So as a breakdown into the structure of the platform, so we have the WebPlay format. As you can see, now we have an examples of the 96 WebPlays. And with that, we connect multiple wells together as a microfluidic unit. And we have the inlet and outlet connecting the bottom microchannels. With that, we aim to have we induce the flow into the microchannels, and then we try to mimic the blood vessel with endothelial cells on the bottom. And then on top of the microchannel, we have the membrane, the porous membrane, where we can then have another cell type on top of that, with the confined inside of an open top chambers. And with that, we now have the flexibility of having both the flow and the static culture in the same testing unit. Because why do we aim for that? Why don't we just have the flow everywhere? Because, for example, in the blood-brain barriers model, then we have the blood vessels, and then the perivascular structures like astrocyte inside the brain. There's actually no flow. So in that way, we can mimic, our platform can be more versatile. And also the unique selling point here is that we have high throughput platform, like multiple testing units per place. But then if we want to induce the flow, imagine if we have pumps, then we have like dozens of tubings have to be connected here, and then multiple pumping system have to be connected here. And this is not a good solution at all. So that's why we use the gravity-induced flow based on a fully programmable rockers that we develop in-house as well. With that, you have the full control of the flow, shear stress, pressures, and I will also go in a bit more detail in the later stage. With that single plate, with that single concept, what we can do, we can have single organ model on a chip, which we can increase all the way up to 128 tests per plate, or you can also connect multiple units together. And then you can have multiple organs on a chip to study, metabolism, cancer metastatics, and drug response between organs, organ-organ interactions. And moreover, we also have the electrical readout, which is the impedance spectroscopy, which in the end we can induce the values of the trans epithelial electrical resistance of the cell layers or the barriers, confluency, for example, or barrier integrity based on this kind of readout. And the device also what we make in-house, and we have is also in the high throughput format. It is compatible with our Akita platform, and also it's reverse compatible with the conventional 24 or 96 transfer insert plate as well. So yeah, this is and also So in terms of the biological validation, what would be the benefits that we are bringing with the Akita platform? So this is just one quick, recap about the capacities of the platform. I will go into in more detail with all the models. We have the blood-brain barriers that we can improve the barrier tightness, allow for small or large molecules crossing the barriers, and also try to approach the in vivo values of the permeability of the barriers, and increase the barrier maturation and polarization with the platform thanks to the flow, and apply later on for mimicking the drug resistant in vitro with patient-derived organoids or cancer on chips, and also to speed up the maturation time for the experiment as well. Our capacity, we are offering now the Akita plate itself, like I just showed you, or also a ready-to-use kit where you have the plates and the cells of interest and different reagent coating solutions to make sure that you can have the good result based on your research. And we was also doing some CRO services in-house. We provide the model, the platform, and then test the compounds of interest for you. And we are also very open for co-development, publication, and brief summaries of our assays that we are performing in-house or with collaborations, including the barrier integrity with our impedance spectroscopy, or through the fluorescent probes like Dextran assay, for example, PKBD, automatized imaging like confocal imaging, high contact screening, immune cell migration, and tumor metastatic through fast microscopy. And we with the platform, you are also able to detach the cells later on and do RNA-seq or transcriptomic. And also we have internal pipeline for bioinformatics to do fully some advanced omic analysis as well. So we are working with multiple partners all over the world, from Japan, South Korea, India, with CRO, pharma, United States, European as well. And we are certified with ISO 9001 for our production. And so we, like I said, we are very open for scientific development. We had collaboration with other partners. Our work was also published out there. So now I will go into a bit more details about different models that we was developing with the Akita platform. So with this single concept, then we have, you can see here in this map, we have different models and different possibility as well. I will just go briefly through this one because I already mentioned about this. So the first one was also the most recent work that we have done with biosurfaces, trying to integrate biosurfaces, biodegradable and biospun membranes into our platforms. So like I mentioned before, we have the critical part of the platform was the membrane, and usually we use the PET membrane, which is the top left pictures, as you can see here, with 3 micrometer pore size. And you can see already in the pictures, the pores are not homogeneous, not conform. Their size are quite random as well, even though the manufacturer confirmed that it was 3 micrometer, but some holes were merged together and they are not straight inside, but sometimes the hole was tilted as well, and it's also a bit challenging for the cell to really adhere to the surface. And we, with biosurfaces products like biospun, like BDO-GA, BOA, or biospun PU, we was able to really see that we can have a more reproducible kind of membrane to help for the cell to adhere on and to further improve the integrity of the cell layers. So how did we do that? We make a quick test with the culture cell culture on our platform to form the get on chip model. And as you can see here, we tested multiple different conditions. With the control is our conventional PET membrane, which you can find in conventional transfer inserts. And so normally we have to use fibronectin collagen, for example, to do the coating first, to make the adherent site for your cells to adhere on. So so we try to do that also with the biospan membrane, and also we have the no coating condition as well. And with that, we see that for PET membrane, of course, with fibronectin it works the best. With collagen, no coating condition, there's no cell at all that can adhere to the membrane over the course of 14 days. And interestingly, with the biospan membrane, like I saw here, the polyurethane and also biodegradable membrane, all the condition was working really well. The cell was fully confluent, and they even do not require any coatings in order to form a confluent monolayers of cargo cells. So this is also already a huge improvement in terms of the handling, right? And also increase the repeatability of the model that we want to develop. And we didn't just stop it with the immunostaining, but before that we was also doing the permeability assay using dextran molecules. Here we have the 0.4 kilodalton dextran molecules that passing through the membrane. So the way we did that is that we loaded the dextran molecule through the microchannel on the bottom, and then we sample the media from the top of the membrane. So in that way, then we can see how much of the dextran molecules that can pass through the barriers or the gut barriers forming by the cargo cells. And so with different coating conditions, then you can already see that the biospan membrane results in a very consistent permeability levels. And for the PET membrane, already at day 14, you can see that it's a huge strike in the permeability level, and it starts to be quite leaky, and especially when you look at the collagen 1 coating condition, then the PET membrane does not perform well at all. So here, the takeaway message is that with biospan membranes, it doesn't require any coatings. So by using that into our platform, we can ensure the ease of use for the end user. We have lower variability. So this is already a huge improvement for our users. And I'm really excited to see the first step of integrating the membrane and also have the advancement of other models of our portfolio as well. And further details, you can also look at the application notes that we just published together with Biosurfaces a few weeks ago. Back to the organ list, I can already go to the next model where I want to show you the blood-brain barriers, which is with low molecular weight compounds. So the idea is the same as I mentioned before. We have the endothelial cells covering the bottom of the membrane inside the microchannels, and with that we are already able to perform the polarization assays to see really if the cells are forming a barriers here, and we can already confirm the polarization of the iPS-derived cells that we have our own iPS-derived cell line in-house, and we was able to confirm the polarization of the model, and also further step with dextran permeability assays, and with tear measurement we was able to reach up to averagely from 1.5 kilo ohms per square centimeter and all the way up to 2.5 kilo ohms per square centimeters, which is a good tightness for a blood-brain barrier model. And with higher molecular weight compounds, what did we do? So you can see here we have like the mouse astrocyte cells on the top of the membrane, and on the bottom we have the endothelial cells. Sorry. And with that setup, you can see on the right side we have the immunostaining of the multiple of the different cell lines from the top and from the bottom to confirm the cell monolayer. And also we have the TEM images confirm the barrier formation and the cell-cell junction formation as well. We was optimizing the barrier tightness through the fluorescent spectral assay. So we have multiple conditions without cell, with only astrocyte, with mouse brain endothelial cell only, or with the co-culture of multiple cell lines together, like forming a stack on top of the membranes. And with that, we was able to compare with the astrocyte with in vivo values and between the in vivo values, the mouse model on chip and also human model on chips. And from this work, we was able to upload the in vivo values. Why? Out there, the other models, microfluidics based models or in vitro models through the transfer enzymes, for example, the variation was huge and also not that close to the in vivo at all. We took another step to advance the model or to validate the model by trying to open the barriers through using a hyper osmotic solution to really shrink the cell area and enlarge the cell-cell junction. And by that, we was able to open the barriers of on the chips. And also, when we refresh the cell culture media with the fresh media, then the barrier was able to recover its initial state. And from this graph, you can already see that before and during the barrier opening, we have a strike in the permeability of the cell layers. And we confirm that with both the low and high molecular weight Dextran molecule. And the next one is the lung model on chips. The interesting thing with this is that we was able to create the air-liquid interface, which is really mimicking what is happening in vivo, right? So we have the cells, the A509 human alveolar cells on top of the membrane inside the open top chamber. And on the bottom, again, we have the endothelial cells with dynamic flow to our rockers. And with air-liquid interface, we was also able to confirm the tightness of the barriers that were enhanced compared to the submerged condition. So the way we induce the air-liquid interface is that we just simply remove the media from the open top chambers. Of course, we have to also have a cover on top of the open top chamber to avoid the liquid from the bottom to go up to the top, but the handling is fairly simple. And surprisingly, through the SEM image, we are able to see that the cell even form the microvilli structures on the cell layer as well, which is already surprised us with a simple model. And yeah, from the immunostaining pictures, you can see that with flow, we are able to form the mucus on top of the cell layers with this very fang or even not happening in the static culture without any flow. And the gut on chip model again, this is the one that we perform with only the PET membrane. So like long before we had the biospan membrane, and we also confirm it quickly with the Dextran assay and also polarization. And we was able to, with the optimized coating and cell culture procedures, we was able to achieve a faster colon on-chip polarization and maturation of this model within 6 to 10 days compared to 21 days for the state-of-the-art transfer inserts. And this is also a very interesting model that we collaborated with our partner at the University of Eastern Finland. And this is a bit, this is still a ongoing project, so I cannot share much. But in here, during the COVID time, our partners was developing this model based on our platform to have the human nasal epithelial cells and the neuron cells on the two sides of the membrane, and from this we form the nasal epithelium on-chip model to test the sprays based, COVID-19 drug or the vaccine. And so this is a collaborative work we have done with Ossiform. They make the in vitro bone models where we have the 3D printed bone scaffolds, and we was able to integrate that into our platform the same way that we did with biosurface membrane. And with that, we also achieved some better, result when or the handling, improve the handlings of the cell culture models, or have to remove the background noises, improve the signal to noise ratios for the readouts of the fluorescent signals, and stabilize the growth of the cells as well inside the scaffold. And when it comes to cancer metastatics, we, beside the transfer-like model that I just showed you, we also have other product line in-house where we have the microvessels, where we can fine-tune the stiffness inside the micro channel as well. And we are able to make different sizes of the channel to mimic the vessel with different shear stress levels. And with this vessel-like, we not just to apply for the mimicking of the blood vessel, but we was able to mimic the prostate cancer metastasis. And with our publication last year, we was having the model where we had the bone marrow tissue osteoblast culture in the chip, and then we had the prostate cancer metastatic cell like PC3 invaded into the cells, and then with that we was treating it with the DTX inhibitor and see how the cells was fighting against the drugs. In the prostate cancer, without the ASIC knockout comparison, we have the here that we can see that the ASIC knockout confers the DTX chemotherapy resistant, but it's synthesize again the anti-metastatic efforts of the DTX, and we're also able to confirm the cell viabilities over the course of 21 days as well. So again, the same setup. Then we can also have the blood vessel on chip. We can fine-tune the stiffness of the ECM surrounding the microchannel where the cells are adhering on, and with that we can observe the different morphological differences of the cells under the flow and static condition as well, cell orientation, cell area, cell alignment. So all of those matters we are able to check the images like in high resolution, and with our image analysis pipeline that we developed in-house, we was able to find ways of striking differences between the conditions. And so we have the last model, which is the skin model, where we have the same setup where we have the porous membrane, fibroblast, keratinocytes, and with that we have the tear measurement to scan through the whole place at once within 1 minute. And we can also do long-term measurement of tear for that, and we correlate that with the fluorescent probe using the electron assay. Here we use low molecular weight as well, and the T values were also able to use to confirm the permeability or correlate between the fluorescent value as well. With that, but the thing is that with this model we didn't have the endothelial cells on the bottom, and that explains for the decrease of the permeability when we induce the bilateral flow in the microchannel, because in that, like I mentioned, the endothelial cells is supposed to expose under flow, but the other cell type, they mean to be stuck in the static conditions. So by having enough components or cell components, then I believe that we can form a better models for this. And also with different, we also confirm the higher polarization or maturation of the skin with low calcium ion condition by the confocal acquisition of the different markers here. And yeah, so this would be almost everything that I wanted to introduce to you today about our platform. And we are very open for collaborations or support you to develop the models of interest. So please feel free to ask any question or contact me through the email. Thank you.
Thank you, Tuan. Appreciate the presentation on the cutting edge technology that FinAdvance is creating, and appreciate you presenting today. We have a couple questions for you, so I'll read those off to you right now. The first question is, have you done transcriptomics on cacao cells grown on the different scaffolds? What are the main differences in gene expression?
Yeah, so for the transcriptomic on the scaffold, we haven't done that, but our collaborator did. We cannot say any data here, but the point is that because the thing is that in the model you have multiple cell lines interacting with each other, and you have the membrane inside in between to separate them, and you are able to withdraw the cells in the end to perform transcriptomic analysis. And our collaborators or customer have the possibility to do that. And for example, just a few months ago, a collaborators from the Duff Institute in Brussels, they performed the blood vessels on chip using our platform using the patent cells, comparing the patient-derived vascular anomaly cells, and they was able to detect the cell from the chips after exposing to the flow thanks to our platform, and from that they were performing transcriptomics and get the result out. So yeah, so this is fully possible on our platform.
Excellent. And then a couple more questions. One is, what do you envision will be your most requested tissue model, and what types of groups have you been showing the most interest so far?
Sorry, can you repeat the question, please?
Yep, no worries. The question is a two-part question. What do you envision will be your most requested tissue model, and what types of groups have been showing the most interest so far?
Okay, so so far we have collaborators asking for the blood-brain barriers model, which is the the most advanced one, and they are really waiting for the confirmation of that one. And also the cell migration model, where we have I didn't show here, but we have also the migration of the immune cells through the vascular barriers, and for the treatment of cancer, like chemotherapy, for example. And that is also something that is very, very demanding at the moment. And our customers include, academic groups, CRO, pharma. We have collaboration with Japanese, and then we have some paid CRO customer as well. And really, they want those models to be fully developed and for their drug screening or compound screening applications.
Okay. The last question I'm seeing right now, and anyone, if you have any further questions, I will put every presenter's contact information in the chat or the Q&A at the end of this. But the last question I see is, what do your current customers say is the best feature of this platform?
The thing that people really like about the platform is that the platform is really versatile, and you can see that with only one design, you can already have multiple setups for different applications, and you are able to do really the sampling of the cell culture media for time point assays, or even do fixing for endpoint assays. You can do electrical readout, you can do high content screening, and also because we have the platform in the WebLab format, so it's fully compatible with any microscope in the market. You can also have that for robot liquid handling as well. So we have tested our platform with several robotic liquid handling, and it works like seamlessly because anyway it's just a WebLab format, So it really is a scalable platform that the user want to have and to perform the high throughput screening. Yeah.
Great. Okay, I think that's all the questions that I'm seeing right now. If I get any more questions, I will be sure to send them over to you. And like I said, I will add both MJ and Tuan's email information if you guys have any further questions. If you kind of missed a part and you want to go back and look at it, that will also be on the website that you guys can go view again. So yeah. Thank you, Tuan, again for presenting. That was excellent.
Thank you. Thank you.
And our last presenter of the day is Patrick Hayden. His title for his talk is Next Generation In Vitro Human Tissue Models Based on Novel Electrospinning Scaffolding. So I'll let you take over, Pat. Thank you.
Okay. All right. Thank you, Cam. Yeah. All right, thanks, Cam, and hello to everyone who is joining us today. It looks like we have at least a couple of different time zones, so good morning or good afternoon, whatever the case may be. All right, let's see here. Before we delve into the main part of my talk, I want to take a moment to consider the overall landscape of in vitro tissue culture methods and platforms, and consider where biosurfaces electrospun scaffold products fit into the overall picture. So there's a broad consensus now regarding the need to move from traditional submerged monolayer cultures to human models that reproduce the 3D structure and function of in vivo human tissues. And a variety of platforms have been developed to achieve this. We have, for example, the transwell cultures that we're going to talk a lot about today, including the ones that we use, that we incorporated our electrospun scaffolds into. This model has some benefits. It allows wall-to-wall coverage of, it's really good for barrier tissues. So you have access, independent access to the apical as well as the basolateral compartments for assaying the different compartments independently. We also have organoid cultures, which are really popular. Typically, these are done either in submerged or as embedded in hydrogel or some kind of other matrix. We also have this is an example of an aviolix chip. It's kind of like a transwell in that it's a membrane base, but this is really a pretty cool setup where they have actually a dynamic membrane that can kind of stretch and breathe in and out. And they kind of specialize only in the lung in this case, but this is a pretty novel and interesting platform as well. In addition, we have like completely scaffold free. This is the Momentus platform that uses the waveguide technology. This gives both flow and shear force. It's very miniaturized, which can be pros and cons to that as well. But very nice model and useful for a variety of different applications. And then, of course, we just saw Tuan gave us an excellent talk about the Akita platform, which is also incorporates flow and shear force, and is a kind of a mid-size kind of tissue, which is kind of nice, again, depending upon your application. Some of these really super high throughput models, the amount of tissue is so small, it's you may not get that much material to work with or that much media for the assay component. So this is kind of a nice mid-size kind of flow model, as well as additional flow models. For example, this is from Kirkstall, Quasi Vivo. And then finally, the model from the Wyss Institute, which was probably has gotten most of the press, a lot of press recently with their organ on a chip, and they also have a stretchable membrane. The whole chip is made out of PDMS, but this also incorporates flow as well as shear force and dynamic stretching. So what I'm going to focus on today is how we have used electrode spinning technology to open up a new dimension in traditional insert technology that allows development of more in vivo-like and relevant human tissue models. And we'll also touch on how these models can be incorporated into some of the existing flow platforms. We already saw an example with the Akita, and there are other possibilities as well, including other platforms like Quasi Vivo that you can just plug in tissue culture inserts in what we call like a plug and play kind of situation. Okay, so with that, this slide elaborates a bit more on the drawbacks of 2D culture systems compared to the 3D environment. For example, in 2D cultures, we don't have any soluble gradients present on either plastic sheet or even a like a traditional polyester film-based membrane that has trackage pores. We don't have good gradients, and in addition, the cells are just forced into a one-sided polarity. They're only attached on one side, and the apical side is just facing the media, which is not always the normal situation. And again, we just have one continuous thin layer of matrix, and this gives just totally unconstrained spreading and migration in the XY plane. And these adhesions are just restricted to the XY plane. And furthermore, these substrates tend to have a really high stiffness, which is far more stiff than the in vivo matrix that most cells are used to growing in. So in contrast with the 3D environment, we have nice soluble gradients present. There's no prescribed polarity. The cells can attach on any side or multiple sides in a more complex fashion. The matrix is composed of discrete fibrils, and of course this gives there's some spreading and migration is sterically hindered. It just can't just spread forever because it's locked in multiple directions by the matrix. And then again, the adhesions are distributed in all three dimensions. And of course, this is very important because these adhesions, send signals into the cell, so they know what they're binding to. So the cells that are cultured in this two-dimensional, very stiff kind of forced polarized environment are going to behave abnormally compared to how they would normally behave in a 3D environment. So 3D environment is very important. Okay, with that then we'll move on to our agenda. My talk for today will begin with a brief presentation of the electrospinning process, as well as the currently available biospun scaffold products and formats. And next, we'll spend the bulk of our time describing a variety of tissue models that we have been working with, including endothelial, pulmonary, dermal, and gut, and others. And finally, we'll end the discussion with a discussion of future directions that we envision for our electrospinning scaffold technology, including developing additional organs, tissue types, co-cultures, and then, of course, organ on a chip or flow platforms, such as the Akita platform. Okay, then, well, let's begin with the electrospinning process. So we start with a polymer solution, and this is contained in some type of syringe or extrusion device. And when the solution is extruded, while we apply a high voltage field between the solution and some kind of a grounding collector plate, this the nanofibers form as the solvent evaporates and the fibers move towards the ground. And the result is a mesh that consists of randomly oriented nanofibers. So the properties of these nanofibers, they can be made from a wide variety of materials, and we can control, their different properties through a number of different, controlling a number of different variables, such as the concentration of the polymer, the molecular weight, conductivity of the polymer and the solvent, the viscosity of the solvent volatility, and molecular structure. Some environmental variables that are very important to consider are the humidity and the temperature. So it's very important to have a stable environment, because for reproducibility, if you're trying to make a electrospun fiber on a hot, humid summer day, and you go back and try to make the same construct on a cold, dry winter day, you could run into trouble. So you want to really control the humidity and temperature very carefully. And then some other electrospinning variables are the distance between the polymer solution and the collector, the voltage, the flow rate, and then, of course, the nature of the collector. So the next slide is just going to show kind of an animated version of what I just kind of described to you. See if it will play properly. Okay, so now, as we mentioned, this process results in formation of a nanofiber mesh, which is kind of shown here in the middle photo, compared on the left to the body's natural extracellular matrix. And we can see that the nanofiber mesh much more closely resembles the natural 3D scaffold compared to on the right side, this is just an example of a polyester film-based scaffold with the track edge pores, which is the industry standard. So again, compared to the typical industry standard, these electrospun meshes are far less rigid. They allow the cells to interact more naturally, and they induce much less stress to the cells and tend to not induce a very strong inflammatory response, because as the cells bind to these, as we showed in the prior slide with the 3D attachments, they sense that it's a normal environment, and as opposed to excessively rigid or stiff substrates that they spread out, they kind of view this as a foreign thing, and they want to form like a fibrotic capsule or mount an inflammation response. So the electrospun scaffolds have a lot of great properties for use in producing 3D tissue models. All right. So Biosurfaces has developed a pretty wide variety now of biospun scaffold products that make up our line of what we call our in vitro research tools. And these include, starting on the left, 96 well, high throughput transwell permeable supports, as well as 24 well, high throughput transwell permeable supports. And these are based on the plate format that is marketed by Corning. We're partners with Corning. We have a purchase agreement with Corning. They provide the plastic component, and then what we do, we put our scaffold on. We obtain the plastic components without the traditional industry standard membrane attached. So they're just empty plastic components, no membrane at all, and then we attach our electrospun scaffolds in place. And in addition, we have different sizes of individual inserts, and these are they're shown in the inverted orientation, just to highlight the scaffold portion here. Obviously, in the background here, we have the receiver plates, and when we're actually using them, we flip this over and it sits inside the receiver plate, and then on top of that, there's also a lid. One final product that we have electrospun scaffolds that we just can put into the bottom of just a standard tissue culture plate, whether it be a 6 well or a 24 well or different various sizes. And these are held in place with a little O-ring clip that kind of holds them in place. So you can grow cells, say, for example, if you don't want, if you don't need, say, a barrier model you want to access the top compartment, the bottom compartment, you just want to grow your cells in a 3-dimensional substrate, you can use this format and easily remove this clip and remove your cells and material if you want to remove them, or lyse them, get the protein out or the RNA or whatever. So, so yeah, I think the next slide just kind of shows a little higher magnification of the 96 well and the 24 well size, as well as different individual sizes of 24 well, 12 well, or 6 well. And these are the scaffold plates where the scaffolds are just placed into the bottom of standard plates and tissue culture plates and held in place with this little plastic clip that's easily removed. All right. Okay, so the different kinds of materials that we have are fairly varied as well. At the top here, on the left-hand side, we have our degradable scaffolds, which are produced from polylactide glycolic acid. And these come in either 12 or 6 micron thicknesses. And these are just a mix, 50-50 lactide glycolic mix. And an important parameter here is the intrinsic viscosity. So this particular blend has an intrinsic viscosity of 1, and this relates to basically the length of the polymers and will kind of influence how quickly they degrade. We have another version that's same material, 10 micron thickness, with a lower intrinsic viscosity. This one's going to degrade a bit faster. We also have bilayers made out of a mixture of the 1 and the 0.65 intrinsic viscosities, which will kind of give you, blended properties. Part of it's going to be degrading a little bit faster, and you'll have a more open mesh that's still remaining that will last for a longer period of time. And then likewise, a similar scenario was where we mixed the PDLGA with polylactic acid, and which this has a higher intrinsic viscosity and also has a much slower degradation rate. So this kind of scaffold will give you an example where you might want, and it's kind of like 2 layers usually, that we have the PLLA on the bottom and the PDLGA on the top. And so your top layer may degrade relatively quickly, but yet you still have some structure to hold the tissue together for a longer period of time as well. And then finally, we have just a fully PLLA based scaffold. And even though, this is generally considered to be degradable, or biodegradable, but this one's going to last for, a much longer extended period of time compared to the PDLGA. And then on the right-hand side, we have the non-degradable scaffolds. They're used for certain applications, including, the polyester that's available in a variety of thicknesses from 50 up to 150 microns. And these are, I'm going to be showing some examples of these that they have a great utility for making full thickness tissue models. We use this polyester matrix to form the dermal component, or the subepidermal matrix component. And this has a lot of great properties for that. I'll be showing more about that in upcoming slides. And then finally, we have the polycarbonate urethane membrane. This membrane has some scaffold, I should say. This scaffold has some nice properties in that it's not degradable, but it also has some flexibility and elasticity to it. So this, for example, we're evaluating it for use on kind of like a breathable. I showed you in the prior slide, the alveolus chip, where it's kind of can flex and breathe. So this is a urethane kind of membrane. We think they're going to have utility in situations like that. In fact, we're actually working on that one as well. Okay, so the target customer for our products tend to include toxicologists and biomedical researchers looking for alternatives to animal testing and new approach methodologies, or they just want to go 3D and have a human model. As we kind of stress the importance of doing that these days. And this includes, pharmaceutical companies, contract research organizations, government research organizations, as well as academic laboratories. Okay, so now we'll switch gears and take a look at some of the tissue models that we've been working on. And just a bit of an overview here. This slide just kind of shows the tissue model, which the main attributes, it must reproduce the in vivo organ function to be useful to the researcher. And then on the right-hand side, some very specific applications that the customer needs. And of course, the model has to reliably reproduce in vivo-like results. So starting off at the top, we have the skin model, and I'll show you again. We're making a full thickness skin model in our laboratories now. And some of the applications that this is going to be really useful for are skin irritation and inflammation, chemical penetration, drug delivery, toxicity, phototoxicity, drug effects, skin aging, photo aging and wrinkles, wound healing, and specific skin diseases. For example, atopic dermatitis or psoriasis, etc. Ocular models are also possible. We haven't been actively working on the ocular yet, but they're definitely a possibility. MJ did hit on one of the models that he's been working on in his lab that used our biodegradable model. And the applications here are ocular irritation and toxicity, drug delivery, and ocular disease, such as dry eye or macular degeneration. Lung models are also of high interest and high need, both partial thickness and full thickness, including the pulmonary fibroblasts, which are a very important component of the lung epithelial tissue, and also possibly endothelial cells and other cell types, including maybe immune cells. And the applications here include respiratory toxicity, drug delivery, respiratory infection, and specific respiratory diseases, for example, asthma, COPD, or idiopathic pulmonary fibrosis. Vascular models are also of high interest, including the blood-brain barrier. Tuan just mentioned that they see that as one of their most sought-after models, so this is a very important one. Intestine is another very important model that's in demand for drug absorption from oral drug delivery, which is ideally, rather than having an injection, if we can take a pill, that's the best route, and we have to test the ability to be absorbed by the gut. We have to test the toxicity, first pass metabolism. Other applications include infection and specific intestinal diseases. Liver is kind of it's not the typical model that we typically use on the inserts. It's not really like a barrier tissue, but I certainly expect that these three-dimensional scaffolds could have utility for development of important models such as liver. And of course, this is the key for drug metabolism and toxicity and liver fibrosis. And then finally, the kidney models, which are in high need for nephrotoxicity and drug development studies as well. All right. So now just move into some of the data that we have. This slide really is just shows a survey just to demonstrate that all of our scaffolds, the PDLGA, polyurethane, and the PET, all of them are actually very good substrates for a wide variety of cells. And here we have just incorporated, this is a cell line, an MDCK cell line, canine kidney cells. Here we have human coronary artery smooth muscle cells. Here we show human coronary artery endothelial cells, and here normal human dermal fibroblasts. So we can see this is stained with either the Hook's dye, staining the nuclei, or phalloidin. And all of these scaffolds are able to support the attachment and growth of a wide variety of these cell types. All right. One more specific barrier model that we have been working with is the endothelial barrier. And we developed this model for evaluating mechanisms for drug disease, drug screening, and in this case also, this was during the peak of the COVID, as a potential model to evaluate coronavirus transition across the endothelial barrier. And here we use the pulmonary endothelial cells that were cultured on a very, very thin 10 micron, 10 to 12 micron PDLGA scaffold. And we see we form a nice thin monolayer. The red, actually, we stained it for the presence of the ACE2 receptor, which is actually the receptor for COVID-19. And the green staining shows the CD31, which is the marker for the endothelial cells. And then, of course, we have the Hook's dye staining the nuclei. We've also made a more complex version of the vascular co-culture model that includes both in endothelial cells as well as vascular smooth muscle cells. And again, this was for testing, disease progression and drug screening applications. So here, these were coronary artery endothelial cells as well as coronary artery smooth muscle cells, again, grown on the very thin PDLGA scaffold. And the thin scaffold, what we did here, we put the endothelial cells on one side and the smooth muscle cells on the other side. And this very thin biodegradable membrane, especially with time, really allows much better than the traditional track edge pore membranes, allows really good cell interactions to form. So we see here the staining of the alpha smooth muscle cells on the bottom, endothelial cells on the top, as well as the nuclei shown. All right, this was a really interesting model of the alveolar barrier model. And again, we were kind of developed a lot of this work was done, during the height of the coronavirus when it was just coming on board. So we kind of were, focused on coronavirus transmission. In this model, again, we used, a very thin biodegradable PDLGA scaffold, and we used human pulmonary microvascular endothelial cells on one side, and then we cultured human alveolar epithelial cells on the other side. And we showed, this is kind of a composite. You can see that they actually, when these two cell types were together, they really formed a really kind of unique three-dimensional structures, and the way they interacted was quite unique. What I don't show here, but we actually did have very high tier, showing that we did have a fully intact alveolar barrier. The staining here shows the ACE2 receptor as well as the CD31 and the hook styrus for the nuclei. All right. So this slide, I just wanted to take a minute to show you, how we handle seeding of the two different cell types, one on the bottom and one on the top. So on the left, this just kind of shows how you would seed the human pulmonary microvascular cells, for example, or whatever the cell may be. We simply flip over the scaffold plate, and you can take a suspension of your cells and then just pipette it on the bottom. This can be done either manually. I just kind of did this by hand with an electronic pipetter. I just one by one, just dispensed one drop, about 25 to 50 microliters, which just kind of sits by surface tension on the underside of the scaffold. Or you could do it robotically or with multi-channel pipetters as well. And so after plating these like in this fashion, you just let it sit. Generally, I just leave it in culture hood, close the sash, and turn the airflow off so it doesn't evaporate, and let it sit for half an hour or so. And that's generally an hour at the max. The cells will generally adhere quite well during that time frame. Then you flip it back over into the reservoir plate with media, and then you can seed your other cell type, in this case, the epithelial cells on the opposite side of the scaffold. And the other thing I wanted to show in the right-hand photo is the use of this height extender. So often when we do air-liquid interface cultures, we want to remove all the medium from the apical side and put it only in the bottom, but the wells really don't accommodate that much medium, and we like to be able to give them more medium. We don't have to feed them as often, and then cultures often require more medium to develop properly. So what we do, we just created this very simple 3D printed little extender made out of plastic that we just put between the reservoir plate and the scaffold plate, just to kind of shim it up a little bit so we can get more medium underneath of it. And that works quite well. We do that with the 96 well as well as the 24 well when need be. And we have a different way to using this hanging top, which I'm not going to really show here, or it can be used if you have individual inserts and you need to raise those up to do air-liquid interface culture. Okay, another very interesting thing that we did with this alveolar model, we evaluated and kind of developed a pulmonary inflammation model using the alveolar co-culture, and to model, for example, exposure to a virus, for example, the COVID vaccine, COVID virus, and exposure to viral particles or other inflammatory stimuli, generally even from the innate immune properties of the epithelium alone, causes release of a variety of cytokines and chemokines, such as IL-8, IL-6, IP-10, and as well as barrier disruption. And then these in turn, so this is the initial inflammatory signal. This attracts in your dendritic cells, your macrophages, your monocytes, and this can lead to the, what we've heard about the cytokine storm that is, implicated in a lot of the deaths caused by viral infections with COVID. So we wanted to see if we could kind of model this. And so what we did, we challenged our tissue with either some known inflammatory stimuli, including TNF alpha, interferon gamma, or combinations of TNF alpha and interferon gamma, as well as poly-IC. Poly-IC being a piece of double-stranded RNA that mimics viral particles or to mimic viral exposure. So in our first pilot experiment, we did find that compared to the unchallenged, that exposure to TNF alpha or a combination of TNF alpha and interferon gamma or poly-IC did indeed cause significant lowering of the barrier and concomitantly pretty robust secretion of IP-10 as well as IL-8. In subsequent experiments, we evaluated a little bit further, looking at the dose response, and we found in this case that it was primarily TNF alpha that led to the disruption of the barrier, as opposed to the interferon gamma. Maybe even a little bit of synergy when you put the TNF alpha and the interferon together, but TNF alpha did it by itself as opposed to interferon gamma. On the other hand, TNF alpha didn't cause too much release of IP-10, whereas interferon gamma did. It's no surprise there. IP-10 is interferon-produced protein 10. So true to form, we saw a very strong, robust response to in terms of IL-10, IP-10 secretion. And then the combination of TNF alpha and interferon gamma may have been slightly synergetic or at least additive here. And then finally, we looked a little bit at IL-6 as well. And in this case, we found that it was TNF alpha that elicited the secretion of IL-6. Not so much for interferon gamma, but it does look like somewhat of a synergy between the TNF alpha and the interferon gamma in producing IL-6. And then finally, poly-IC also led to the induction of IL-6. So the conclusion then here is that the biospun alveolar culture tissue model it's a useful model for modeling pulmonary inflammation, and should be useful for toxicity and drug development applications as well. All right, changing gears then here. This slide shows the development of a full thickness airway model. And so, often we see it's quite common to have partial thickness bronchial epithelial models that are useful for quite a lot of applications, tobacco smoke, tobacco research, or e-cigarettes, again, pulmonary infection, just inhaled drug delivery, a wide gamut of needs and uses there. But the bronchial tissue, the fibroblasts in the subepithelial matrix, they plays a big role in a lot of these processes. And so, if you're only looking at a partial thickness model, you're missing a major component. So what we're trying to do now, and this uses our polyester, thicker polyester scaffold, is to create what we call a full thickness in vitro human airway model. And the benefits here, compared to other types of, full thickness bronchial models, that usually they rely on having the matrix is going to be made from some kind of animal derived collagen, whether it be bovine or rat tail collagen or whatever. And aside from being animal derived, these constructs tend to be really, really unstable. They don't have a long lifespan. They tend to contract and fall apart. So the beauty of the polyester electrode biospun scaffold is that we completely eliminate the need for the animal collagen. We put human fibroblasts into the scaffold. Human fibroblasts make human matrix. The scaffold provides structure so that you don't, completely eliminates the contraction problems, and makes a nice full thickness respiratory tissue. So here on the left, we just show like the kind of low mag showing both the matrix as well as the epithelial portion, and then we have some other views of the higher magnification showing, focusing more on just the epithelial portion and just below the epithelium. So here we see the really nice, what we call this pseudo-stratified mucociliary structure of the epithelium, and here we see some of the fibroblasts which have actually migrated out. You can kind of see the polyester fibers here. So this scaffold is like very open mesh, so the cells can easily go through it, which is also great. In future applications, we want to add immune cells, so we think that the cells can easily migrate in and out, which is going to be really important and not so possible with the traditional trackage pore based industry standard scaffolds. We think that there's going to be a big improvement for immune cell migration, as well as just nanoparticles going in and out, drugs, you name it. It's not an unnatural barrier here. So there is the airway model, full thickness airway model. We've also worked on some intestinal models. This work was actually done in collaboration. In the early days, we had a collaboration together with my former colleagues at MatTech, and so this model was put together by Dr. Sayam Maihuni. And in this case, this was kind of a full thickness model that utilized intestinal fibroblasts as well as intestinal epithelial cells. And this was, again, the polyester, the thicker polyester. And we also made a model on the very thin PDLGA scaffold, which included only the intestinal epithelial cells. And in this case, you can see that the cells actually, since this is, again, pretty open and it does have some pores and will begin to degrade and disappear with time, the epithelial cells actually started growing on the underside as well. So there's the intestinal model, and then finally now we come to our full thickness skin model. This is a major application that we're working on now. This going quite well, and is the focus of phase 2 SBIR that we recently were awarded from the NIEHS. And by the way, we're going to be working on developing the full thickness bronchial models under this project as well. But so far, this is the very latest data that I've been able to produce using the full thickness skin. It's similar to what I described for the full thickness airway. We use 150 micron thick PET scaffolds. Same benefits. We don't have any use of animal derived collagen. Fully human derived matrix, completely eliminates the typical issues with tissue stability and contraction. So here on the low magnification view on the top just shows, so this here is the scaffold, and these arrows show where the plastic wall would be. But of course, this tissue has been removed. You can just easily, with a pair of forceps, kind of grab the end and just pull the whole tissue out of the scaffold if you want to do histological processing like we're seeing here. So this just shows very uniform wall-to-wall coverage of the epithelium. And then again, the dermal component is actually inside this, for the most part, this electrospun scaffold. Below, we see three different sections. This is the same tissue at different spots, showing the really nicely formed epidermis, including all the typical layers, stratified basals, spinous layer, granular layer, and stratum corneum with barrier. I'll show you the basement membrane zone in a minute. And here again, we can see the fibroblast incorporated into the scaffold, and then there is this thin layer that's kind of has migrated outside of the scaffold. It's just beneath the epithelium as well. And the thickness of this layer can actually be controlled by a couple of factors, namely, how many fibroblasts you seed, and how long you culture it prior to adding the epidermis. So this model is working really quite well. I can show you here. This just on the left just shows an example of the immunohistochemistry showing collagen 1 deposition. Again, this is completely animal collagen free, secreted and assembled in situ by the fibroblasts. So we can see there's quite copious deposition of collagen 1 throughout the entire matrix. And of course, this is where the epithelium is, and you can kind of see the Hooke's die of the nuclei of those basal epithelial cells at the top. And then here's the basement membrane zone that I alluded to. So very importantly, it's very important to have a good basement membrane of your full thickness tissue models, because the basal cells, they know what they're binding to. They can sense. They have receptors on their basal laminal membranes, and that includes, for example, these hemidesmosomes. And in the back of these hemidesmosomes are tonofilaments, and these signal back into the cell to tell the cell that the state of this situation. It's like, okay, we have a fully covered basement membrane, or okay, we have a wound here that we need to migrate and heal this wound and cover up the area, or everything's happy and healthy. So this is very important to have this to signal back. And not only that, this even, believe it or not, impacts even things like the barrier of the tissue. So here on the dermal side, we see the lamina densa, which is kind of this electron dense boundary here. Here we see the different striated collagen fibers. Again, these were completely produced by the fibroblast, and they self-assembled. And then this is pretty amazing. We can even see the anchoring fibrils. So this is what kind of binds the dermis to the epidermis. This is primarily typically made from collagen 7, but we see these little striated anchoring fibrils that they kind of loop around the lamina densa and loop around these striated collagen fibers. And this is kind of the actual mechanics, the mechanism of keeping the skin attached to the dermis. So that's pretty cool. We have that as well. And then okay, so this slide just gives you a time course of stability and development of the tissue model. We've gone from 3S8A out to 3S28A, and 3S means that basically 3S just means that the model, the tissue was cultured submerged for 3 days before we raise it to air-liquid interface. So still a little bit underdeveloped by 3S8A. That's again 3 days submerged, 8 days at the air-liquid interface. But by 3S13A, it seems very well developed, as can be seen down here in the higher mag. Maybe it's a better place to look. And this remarkably remains. We just took it all the way out to 28 days, and this was we stopped it at 28 days. It was still looking great. It was remarkable. It kind of maintained the very stable epithelial layer thickness. No contraction again, because typically, you have a very relatively short window for use with these other, commercially available or other homemade full thickness tissue models. They tend to fall apart pretty quickly, and as I already mentioned, they tend to have contraction and stability problems. So here we took it all the way out to 28 days. So we got, we have a really nice window of use here, at least from 13A out to 28A, with no appreciable loss of histological, morphological appearance. And then up here, this kind of just also shows the same thing, that, still maintains really nice uniform wall to wall during the entire course of certainly from 13 to 28 days. All right, and then finally, this is kind of out of the realm of the tissue models that I've been really discussing, but another possible utility of the electrospun scaffolds is as a carrier or a substrate for spheroids. A lot of times we'll grow spheroids either in ultra low binding plates or in media, but sometimes you want to move them around, maybe to transport them or ship them, or perhaps you want to develop a different co-culture model of some kind where you actually would like to let the spheroids attach. And so this just shows that the spheroids even can readily attach. This kind of this is a piece of scaffold that was kind of jammed down into the well of a plate. So that's why you can see the kind of sharp corners here. But on the bottom, you see that these were kind of neurospheres that we were working with that attach really well, and we can kind of see them attaching to, in this case, we have PET, different charges of PET. They seem to prefer the negative charge PET. I didn't really go into too much detail about in the case of specifically the PET. We can actually control the charge of the polymer, make it neutral, positive, or negative. So which we have here, and then also the PDLGA. They just really love to bind to that. So that's another potential utility of our biospun scaffolds. And now I think that's all the data I had to show. I want to talk briefly about some of the future directions that we envision. And in the immediate future, I kind of already alluded to the fact that we have a phase 2 SBIR model. So we're actively working on that. And this will include further development and validation of our full thickness skin and airway models. And again, the applications here are for skin and pulmonary toxicology related applications. We are going to be collaborating with the Institute for In Vitro Sciences in this regard. They'll be doing a good bit of the GMP actual testing. We're going to deliver the tissues down to them so they can test the reproducibility and test various applications, including chemical toxicity, phototoxicity, photoaging, airway toxicity, and tobacco research. So that's one big thing that we've got ongoing in the lab. We also envision, as I kind of alluded to before, that a whole long laundry list of potential additional organ systems that we could eventually bring to bear, including the gut, liver, ocular, kidney, vascular, blood-brain barriers, high up on that list, as well as oncology or in vitro cancer models. So lots of potential, more than we can feasibly work on right now, but they definitely are important applications that are of high interest and high potential. And then finally, we do have some active collaborations going on, as we mentioned. Tuan mentioned in his prior talk with the FinAdvance. We've been working together with our partners at FinAdvance to incorporate the electrospun scaffolds onto their Akita platform, and we're really excited about this. We think that this is going to open up a whole very fruitful partnership to develop some really nice models with additional model complexity and functionality, adding endothelial and immune components, and so on. And as Tuan had mentioned, that they have found in their preliminary pilot studies using our electrospun scaffolds that they do have improved performance compared to the standard two-dimensional track etch pour polyester film-based scaffold, I should say membranes in that case. And then one key attribute that seems to be very helpful is that our scaffolds don't require any kind of pre-coating, so it just makes it very user-friendly and easy to use. So let's see, I think that might be it. Yeah, that's my last slide. I want to acknowledge our whole team here at Biosurfaces. Everybody in all the slides I've shown here, everyone had a big hand in doing a lot of work, developing the scaffolds and making the plates and spinning the scaffolds and conducting the experiments as well. We had some collaborators also helping us with our confocal microscopy. Anthony Heng and Glenn Gaudet. They were originally at Worcester Polytech. Now they're Glenn and his postdoc Luke Peralt are down at Boston College doing confocal imaging for us. We'd also like to thank the folks at NCATS. They have been a funding source for us and have been instrumental in supporting us. We're really appreciative of that as well. And then the NIEHS, which is our funding source for our current phase 2 SBIR grant. And last but not least, our partners at FinAdvance, Tuan and Sebastian and his group there at FinAdvance, who are our partners for developing the flow models on the Akita Flow platform. Okay, and so with that, I think that's it. I'll stop sharing and be happy to take any questions, and thanks for your attention.
Awesome. Thank you, Pat. So I'll answer the first one. It was one of them was from Brian asking if Biosurfaces will be attending AAPS. Unfortunately, not this year, but we are definitely looking into attending meetings like that coming up in 2024. So I appreciate the question, Pat. So 2 questions from Oris. One of them is slide 27 is very impressive. Do you have both fibroblast and myofibroblast?
Well, we did not stain the myofibroblast. Usually we would look for smooth muscle actin. I haven't done that yet, so I don't know to the extent that they may have myofibroblast properties. So, but we during the course of our upcoming phase 2 studies, we'll be further exploring those other endpoints as well. So sorry, I don't have the answer for you right now.
Nope, that's fine. And then the other one from Oris, it seems more like a statement, is slide 31 would be good to test for wound healing and scar formation. I don't know if you have any comments on that for slide 31.
Since I don't have it right in front of me, what model was that? If you could ask. Hang on, let me look. Slide 31. For my slide 31 is is future directions. What?
Oris's question maybe, are we planning to look into wound healing or scar formation testing?
Well, the short answer, it's definitely a possibility. I mean, it's not one of our specific aims to look at wound healing, but that is a big interest. I know we have done some work in the past where we've looked at, and you can easily do it, either take a small punch biopsy and make a wound. You can do different kinds of wounding using, for example, burn wounds. Like I've had a little cautery device. You can just kind of torture the little pieces of skin. You can burn them. You can freeze them. You could put like a little frozen piece of like a little piece of metal and put it in liquid nitrogen or something and do a freeze burn. Or you can do chemical burns, which are a little bit difficult because the tissue tends to spread. For example, we've done things, I don't know, very sodium hydroxide or something really caustic. You just literally put a microliter or 2 And pretty quickly, you got to just kind of rinse it off as fast as you can, because even as you rinse it off, it will start spreading. But if you do a lot of quick rinses, you can develop chemical wounds. So yeah, there's a lot of possibilities for wound healing, whether physical wounds, cut wounds, burn wounds, freeze wounds, and different kinds of chemicals. So that's definitely a possibility.
All right, perfect. And just I'll ask a couple more questions here, and then we can wrap it up. How thick can the biospun scaffolds be made?
Yeah, okay, so I think that kind of varies on the different materials. The biodegradable PDLGA scaffolds, they range, thinnest is about 6 microns. I think we can make those up to about 20. The polyurethane, usually we go from 10 to 20 is kind of nominal. I think the highest is up to about 100. We don't generally make them that thick. And then the PET, they probably, range from about 20 on up to 500 or so. In the slides I showed, we used 150 micron, but in the past we've done 200, 300, 400, almost 500 microns thick. So that can be customizable. How thick we want them to be.
And the final question is, how do the 3D scaffolds compare to 3D spheroid models?
Yeah, so I know a lot of people are using the 3D spheroids, and they're great for a lot of applications. A couple of issues with the spheroids, though. Of course, generally they're going to be in submerged culture or embedded into some kind of matrigel or something like that. So it's kind of difficult to dose them, and maybe more importantly, you don't have good polarized compartments. I mean, they're to be polarized, but say for example, since they're circular, say for example, the lumen is inside of the sphere. So how do you access that if you want to evaluate something, or you want to look at drug transport or something like that? So the scaffold technology and the inserts are, I think, in my view, much superior to that, because here you have the polarized, what I showed with the example of the skin and the airway and the other tissues as well. They form wall to wall inside the scaffold. You've got the apical side is completely segregated away from the basolateral side, and so you can have medium on both sides. You can have air liquid interface on one side or whatever. So you can access both sides independently, and you can do nice transport studies and things like that. So I think that's the benefit. I know a lot of people use spheroids, and they're useful for a lot of things depending upon your application, but I think for a lot of toxicology and drug transport and drug delivery and drug development applications, I think that the scaffold technology and the inserts is superior.
Awesome. Thank you so much for presenting, Pat. That was extremely knowledgeable, and I appreciate it. I just wanted to take a quick time to thank all of our presenters today, and thank anyone that has stopped by to view our webinar. This is the first one, and I hope to have another one, and maybe we'll have an announcement soon enough. We did end up taking almost the entire time, so anyone that truly stuck around, thank you for giving us your time. I hope you guys have a great rest of your week, and thank you again to everyone.